Welcome back to the Sleepy Mind channel. Tonight we explore mushrooms, following fungal growth, nutrient exchange, and long-term ecological processes as they unfold over long periods of time and shape the system in steady, observable ways. If you find these long, calm science documentaries useful, you can like the video and subscribe for more. If you choose to leave a comment about where you are listening from, it is always interesting to see how far these stories travel. Let us begin. Mushrooms are almost everywhere, whether they are noticed or not. They exist beneath lawns, forests, farms, city streets, and buildings, extending through soil, wood, and organic material in forms that are rarely seen directly. What is commonly recognized as a mushroom is only a brief surface structure, produced for a short period of time by an organism that may have been present in the same place for decades or longer. Without fungi, many of the systems that support life on land would not function in their current form. Large amounts of plant material would accumulate rather than break down. Nutrients such as nitrogen and phosphorus would remain locked inside dead tissue instead of returning to soil. Many medicines that are now taken for granted would not exist, including antibiotics derived from fungal compounds first identified less than a century ago. Fungi are not plants, and they are not animals. Genetic analysis places them closer to animals than to plants, although their way of living differs from both. They do not photosynthesize, so they do not produce energy from sunlight. Instead, they release enzymes into their surroundings and absorb nutrients once those materials have been broken down at a molecular level. This process allows fungi to access resources that are unavailable to most other organisms. Wood, for example, is composed largely of cellulose and lignin, substances that are resistant to decay. Certain fungi are able to break these compounds apart using specialized enzymes, converting solid plant structures into simpler molecules that can be absorbed and redistributed. The organism doing this work is not the mushroom itself. It is a network of fine filaments called hyphae. Each hypha is microscopic, thinner than a human hair, and capable of extending through tiny spaces in soil or organic material. As these hyphae grow and branch, they form a larger structure known as mycelium. Mycelium spreads outward rather than upward. It follows gradients of moisture, temperature, and available nutrients, adjusting its direction as conditions change. Growth is not random. It responds to chemical signals released by plant roots, bacteria, and other fungi, allowing the organism to prioritize certain areas over others. In favorable conditions, a single fungal individual can extend over a large area. Some mycelial networks cover several square kilometers and are among the largest living organisms on Earth by area. Despite this scale, their presence remains mostly unnoticed because they remain below the surface. Energy movement within a fungal network does not rely on a central control point. Nutrients absorbed in one location can be transported to another through cytoplasmic flow inside the hyphae. This movement allows resources to be redistributed toward areas where growth or repair is needed, supporting the organism as a whole rather than any single point. Water plays a critical role in this process. Fungal cells rely on moisture to maintain internal pressure, which in turn allows hyphae to push forward through soil and organic matter. Changes in rainfall, humidity, and soil structure directly influence the rate and direction of growth. When conditions shift in a way that favors reproduction, a portion of the mycelium redirects its resources upward. Cells near the surface begin to organize into denser structures, eventually forming what is recognized as a mushroom. This structure develops quickly compared to the slower expansion of the network below. The mushroom exists primarily to produce and release spores. Gills, pores, or other specialized surfaces increase the area available for spore formation. Once mature, millions or even billions of spores may be released over a short period of time, dispersing through air currents, water movement, or contact with animals. Each spore contains the potential to form a new fungal network if it lands in a suitable environment. Most do not. Successful establishment depends on moisture, temperature, available nutrients, and the absence of competing organisms. This low probability is offset by the sheer number of spores released. Long before humans began naming or categorizing fungi, these processes were already shaping terrestrial ecosystems. Fossil evidence suggests that fungi played a key role in the colonization of land by plants over 400 million years ago. Early plants lacked complex root systems and depended on fungal partners to access nutrients from mineral soil. That partnership continues today. Many plants form symbiotic relationships with fungi known as mycorrhizae. In these associations, fungal hyphae connect directly with plant roots, extending their effective reach. The plant provides sugars produced through photosynthesis, while the fungus supplies water and minerals absorbed from the surrounding environment. This exchange is not evenly distributed. Fungal networks can allocate resources between different plants, favoring some over others depending on species, health, and environmental conditions. The result is a dynamic system where energy and nutrients move through living connections rather than remaining isolated within individual organisms. At the scale of a forest, these interactions influence growth rates, resilience to drought, and recovery after disturbance. Trees connected to extensive fungal networks often show improved access to nutrients compared to those growing alone. Seedlings that connect early may establish more successfully than those that do not. Even outside forests, fungi continue to shape environments quietly. In grasslands, agricultural fields, and urban soils, fungal activity influences soil structure by binding particles together. This improves water retention and reduces erosion, affecting how landscapes respond to rain and wind over time. Despite this influence, fungi are often perceived only when something goes wrong. Mold on food, disease in crops, or decay in buildings draw attention to their presence. These situations represent the same biological processes that operate continuously in natural systems, applied to materials humans prefer to keep unchanged. Understanding fungi requires adjusting how scale and time are considered. Growth occurs over days, months, or years, rather than moments. Structures appear and disappear, while the organism persists. Activity is distributed rather than centralized, responding locally to conditions rather than following a single directive. As observation continues, it becomes clear that the visible mushroom is not an isolated object, but part of a much larger system, extending beyond the limits of sight. What happens at the surface reflects processes that have been underway below for long periods, shaped by the movement of water, energy, and chemical signals through living networks. The ground beneath these systems is not static. Soil composition changes as fungi break down organic material and redistribute nutrients. Microorganisms respond to these changes, forming communities that interact with fungal networks in complex ways. Over time, This activity alters the physical and chemical properties of the environment itself. These changes accumulate gradually. A single fungal network may persist through multiple seasons, adapting to temperature shifts, moisture variation, and competition. Growth slows during unfavorable periods and resumes when conditions improve, maintaining continuity even when surface signs are absent. This ongoing presence sets the stage for everything that follows. What appears later depends on what has already occurred below. The systems operating here do not rush. They respond, adjust, and continue, shaping the environment through steady processes rather than sudden events. The presence of fungi at this stage is already influencing processes that will not become obvious until much later. As organic material enters the soil, whether from fallen leaves, dead roots, or decaying wood, fungal enzymes begin altering its structure. Complex carbohydrates are reduced into simpler compounds. Mineral elements are released and redistributed. This activity changes what is available to other organisms long before any surface growth is visible. Unlike animals, fungi do not ingest food in discrete events. Their interaction with the environment is continuous. Enzymes are secreted into surrounding material, digestion occurs externally, and absorption follows along concentration gradients. This method allows fungi to feed on substances that cannot be consumed in solid form, including intact plant cell walls. The effectiveness of this strategy depends on surface area. Hyphae branch repeatedly, creating extensive contact with the environment. A small volume of soil can contain many meters of fungal filament. This structure allows nutrients encountered in one location to influence growth elsewhere, as internal transport redistributes resources through the network. Chemical signaling plays a central role in this coordination. Gradients of sugars, amino acids, and minerals guide hyphal growth. When a nutrient-rich patch is encountered, branching increases locally. When conditions deteriorate, growth slows or redirects. These responses are governed by biochemical feedback rather than central control. Temperature affects these processes in measurable ways. Many fungi operate most efficiently between 10 and 25 degrees Celsius, although species adapted to colder or warmer conditions exist. Below freezing, metabolic activity slows sharply. Above certain thresholds, proteins denature and growth becomes unsustainable. Seasonal cycles therefore influence fungal activity, even when soil conditions appear stable. Oxygen availability is another limiting factor. Most fungi require oxygen for respiration, but their tolerance varies. In compacted or waterlogged soils where oxygen diffusion is limited, fungal growth is reduced. In contrast, well-aerated soils support more extensive networks, allowing faster nutrient cycling. These constraints shape where fungi thrive and where they remain sparse. In forest soils rich in organic matter and moderate moisture, fungal biomass can exceed that of bacteria. In disturbed or heavily managed soils, bacterial processes often dominate instead. The balance between these groups influences how quickly nutrients move and how stable the soil remains. Within this environment, fungi interact continuously with other microorganisms. Bacteria consume byproducts of fungal digestion. Protozoa feed on both. Competition occurs when multiple species target the same resources. Chemical inhibition can suppress rivals, while tolerance allows coexistence. The resulting community structure reflects long-term interactions rather than short-term events. This microbial activity contributes directly to soil formation. As organic material is decomposed and minerals are mobilized, fine particles bind together through fungal secretions. These aggregates improve soil porosity, allowing water and air to move more freely. Over time, this process alters root penetration, drainage patterns, and erosion rates. The effect extends upward into plant growth. Roots entering soil shaped by fungal activity encounter a different chemical and physical environment than roots in undeveloped substrate. Nutrient availability, water retention, and resistance to compaction all influence plant performance. These factors accumulate gradually, influencing vegetation patterns across landscapes. At the level of the individual fungus, growth remains opportunistic rather than planned. Hyphae extend where conditions allow, retract or die back where they do not. Older sections of the network may persist, even as new growth explores outward. This creates a patchwork of activity within a single organism, reflecting its history of interactions with the environment. Over time, sections of mycelium age and degrade. Cellular components are recycled internally or consumed by other organisms. This turnover allows the network to adapt without maintaining every part indefinitely. Longevity arises not from permanence of structure, but from continuity of function. When environmental signals align, reproductive structures form again at the surface. These events are not random. They follow periods of sufficient resource accumulation and favorable external conditions. Moisture availability is particularly important, as the rapid expansion of mushroom tissue relies on water uptake rather than cell division alone. The speed at which mushrooms appear can therefore be misleading. Much of the growth occurs through inflation of existing cells, driven by internal pressure. This allows structures to emerge within hours or days, even though the underlying organism developed slowly over much longer periods. Once released, spores enter a different phase of uncertainty. Their small size allows long-distance dispersal, but also exposes them to desiccation and ultraviolet radiation. Many remain dormant until conditions permit germination. Others never encounter suitable environments at all. Those that succeed initiate the same processes observed earlier. Hyphae extend, networks form, enzymes are released, and material is transformed. Each successful establishment adds to the broader system operating within the soil, reinforcing patterns already present or introducing new dynamics. From this perspective, fungi are not episodic features of ecosystems. They are continuous participants. Their activity does not begin with the appearance of mushrooms, nor does it end when those structures decay. Instead, visible forms mark brief moments within much longer sequences of growth, interaction, and transformation. This understanding reframes how presence is defined. What is unseen is not inactive. What appears briefly may represent years of prior development. Time within fungal systems operates on overlapping scales, from rapid surface change to slow underground persistence. As attention remains with these processes, distinctions between organism and environment become less clear. Fungal networks extend into soil, wood, and living roots, blurring boundaries between individual and system. Material moves through these connections, linking separate organisms into shared pathways of exchange. These pathways establish the foundation for further complexity. As networks expand and intersect, they influence which organisms thrive, how resources are distributed, and how the system responds to change. Everything that follows builds on this groundwork, already in motion beneath the surface. The focus now shifts gradually toward what these organisms are, beyond their growth patterns. Understanding their structure at the cellular and biochemical level reveals why these systems behave as they do and how their physical makeup enables such extensive influence over land-based life. At a closer scale, the structure of a fungus explains many of the behaviors already observed. Fungal cells are surrounded by walls made largely of chitin, a strong nitrogen-containing polymer also found in the exoskeletons of insects. This material provides rigidity while still allowing flexibility, supporting growth through narrow spaces without the need for rigid internal structures. Inside each hyphal strand, cytoplasm flows continuously. Unlike plant cells, fungal cells are often not fully separated by solid walls. Instead, pores allow material to pass from one segment to the next. This arrangement enables nutrients, water, and signaling molecules to move freely across large distances within the same organism. This internal connectivity allows fungi to function as integrated systems rather than collections of independent cells. Damage in one area can be compensated for by resources from another. Growth in one region can be supported by digestion occurring elsewhere. The network adjusts dynamically to changing conditions without requiring specialized organs. Energy acquisition in fungi relies entirely on chemical gradients. Sugars, amino acids, and other small molecules move from areas of high concentration to lower concentration within the hyphae. Transport proteins, embedded in cell membranes, regulate uptake and distribution, ensuring that metabolic demand is met where activity is highest. Respiration within fungal cells follows pathways similar to those in animals. Glucose and other carbon compounds are broken down through glycolysis and the citric acid cycle, producing energy in the form of ATP. Oxygen availability influences the efficiency of this process, linking fungal metabolism directly to soil structure and aeration. Nitrogen metabolism is particularly important. Many fungi can absorb nitrogen in inorganic forms, such as nitrate and ammonium, while others rely on organic sources released during decomposition. Some species form associations with bacteria capable of nitrogen fixation, indirectly accessing atmospheric nitrogen through shared systems. Phosphorus, often limiting in soils, is another critical element. Fungal hyphae release organic acids that solubilize phosphorus bound to mineral particles. This process increases availability not only to the fungus, but also to plants connected through mycorrhizal associations. These biochemical interactions scale upward through the environment. As fungal networks mobilize nutrients, they influence which plants grow more efficiently and how communities assemble over time. Small chemical changes at the microscopic level accumulate into visible patterns across landscapes. Growth of the network remains incremental. Hyphae extend at their tips, adding new material only at the leading edge. This allows precise exploration of the environment, responding locally rather than committing resources indiscriminately. Branching frequency increases in nutrient-rich zones and decreases in poorer areas. When resources are exhausted, growth slows or stops. Sections of the network may become inactive without being removed. This creates a living archive of past conditions embedded within the organism's structure, reflecting where nutrients were once abundant and where they were not. Interactions between different fungal individuals occur frequently. When networks meet, outcomes depend on species identity and environmental context. Some merge, sharing resources and forming larger systems, Others compete, establishing boundaries through chemical signaling or physical exclusion. These encounters influence spatial organization within the soil. Territories form and shift over time, shaped by growth rates, resource availability, and disturbance. The result is a constantly evolving mosaic of fungal activity beneath the surface. Disturbance does not eliminate fungal presence, but alters its configuration. Soil compaction, drying, freezing, or chemical input may reduce activity temporarily. Surviving fragments of mycelium often persist and resume growth when conditions improve, maintaining continuity through periods of stress. This resilience contributes to ecosystem stability. After events such as fire, flood, or mechanical disruption, fungal networks often recover more quickly than many other organisms. Their ability to remain dormant and reactivate supports regeneration processes across affected areas. As time progresses, layers of activity accumulate. Older organic material becomes increasingly processed, while new inputs enter from above. Fungi occupy a central position in this flow, linking dead matter to living systems through continuous transformation. The deeper this activity extends, the less visible it becomes. Yet influence increases rather than diminishes. Processes occurring centimeters below the surface determine nutrient availability, water movement, and root behavior over much larger scales. Attention now settles fully on what lies beneath sight. The surface signs that draw attention to mushrooms are transient. The persistent work occurs below, where networks expand, interact, and adapt over years and decades, shaping the environment without announcing their presence. Below the surface, persistence matters more than speed. Mycelial networks remain active, even when conditions are not favorable for visible growth. During dry periods, metabolic activity slows. Water content inside cells decreases, enzymes operate less efficiently, and growth pauses. When moisture returns, activity resumes without the need to rebuild from the beginning. Temperature fluctuations shape this pattern. In temperate regions, fungal metabolism rises in spring and autumn, when soil moisture and moderate temperatures coincide. During summer heat or winter cold, activity retreats deeper into the soil, where conditions remain more stable. This vertical movement allows continuity across seasons. Depth also influences longevity. Hyphae near the surface experience frequent disturbance from drying, freezing, and mechanical disruption. Deeper networks are buffered against these changes. They persist through cycles that erase surface signs, maintaining biological memory within the system. This persistence allows fungi to integrate events over long periods. A fallen tree may take decades to decompose fully. Fungal networks colonize exposed wood slowly, penetrating fibers and altering structure over years rather than weeks. As decomposition progresses, nutrient release follows a predictable sequence, influencing surrounding plant growth long after the original event. Water movement through soil carries dissolved nutrients away from these sites. Fungal hyphae intercept this flow, absorbing minerals before they are lost to deeper layers. This interception concentrates resources within biologically active zones, reinforcing the connection between fungal presence and plant productivity. In this way, fungi regulate not only decomposition, but retention. They slow nutrient loss from ecosystems by holding elements within living systems, rather than allowing rapid leaching. Over time, this contributes to soil fertility and ecosystem stability. Carbon cycling is also shaped by fungal activity. As organic matter decomposes, carbon is released as carbon dioxide through respiration. Some carbon becomes incorporated into fungal biomass. When fungi die, this material contributes to soil organic matter, storing carbon for longer periods. The balance between these processes determines whether soils act as carbon sources or sinks. Fungal-dominated systems often retain more carbon than bacterial-dominated ones, due to differences in metabolic efficiency and byproduct formation. This distinction influences climate interactions at regional and global scales. Despite this influence, fungal activity remains difficult to observe directly. Measurements rely on indirect indicators such as enzyme activity, carbon flux, and nutrient concentrations. The organisms themselves continue operating largely unseen, shaping outcomes that become apparent only through their effects. As years pass, networks fragment and reconnect. Physical disturbance breaks hyphae, but surviving fragments retain the capacity to grow. Genetic continuity persists even as structure changes. This allows adaptation to local conditions without requiring long-distance dispersal. Over decades, species composition shifts. Changes in vegetation alter root exudates and litter input, favoring different fungal communities. Climate trends influence moisture regimes and temperature profiles, further shaping distribution. These transitions occur gradually, layered over existing systems, rather than replacing them abruptly. The result is an environment that carries history within its biology. Present conditions reflect past interactions between fungi, plants, climate, and disturbance. Nothing operates in isolation. Each process builds upon what came before. The surface remains quiet during much of this time. No mushrooms appear. Yet below, growth, exchange, and transformation continue. The absence of visible signs does not indicate inactivity, only that reproduction is not currently favored. When conditions shift again, surface structures may reappear briefly. They mark moments when long-term processes align rather than sudden beginnings. Their presence signals continuity rather than change. Attention now moves forward, following these underground systems as they interact more directly with water movement, plant roots, and the flow of materials across larger scales. What remains unseen continues to shape everything above it. Water moves through soil unevenly. Gravity draws it downward, while capillary forces pull it sideways and upward through small pores between particles. The size and arrangement of these pores determine how quickly water drains and how long it remains available. Fungal networks operate within this moving matrix, responding to changes in moisture that shift hour by hour and season by season. When rain enters the soil, dissolved minerals move with it. Calcium, potassium, magnesium, nitrate, and phosphate travel along water films that coat soil particles. Without interception, much of this material would pass beyond the reach of roots. Fungal hyphae intersect these pathways, absorbing nutrients directly from solution and retaining them within living tissue. This interception alters the timing of nutrient availability. Instead of brief pulses following rainfall, elements are held and released gradually. Hyphae act as temporary reservoirs, storing materials when supply is high and redistributing them when surrounding concentrations fall. This buffering smooths fluctuations that would otherwise stress plants and microorganisms. Water availability also influences the physical behavior of hyphae. Internal pressure within fungal cells depends on hydration. As water content rises, pressure increases, allowing hyphae to extend through soil and organic matter. When moisture declines, pressure drops and growth slows. These responses are reversible, allowing rapid adjustment without structural damage. At interfaces where soil meets living roots, water movement becomes more complex. Roots release sugars, amino acids, and organic acids into the surrounding soil. These exudates alter local chemistry, attracting fungal growth toward the root surface. Hyphae proliferate in these zones, forming dense associations that link plant and fungus directly. In mycorrhizal relationships, fungal hyphae penetrate root tissues or envelop them externally, depending on species. The interface where exchange occurs is highly regulated. Plant cells control the flow of carbon compounds outward. Fungal cells regulate the inward movement of water and minerals. This exchange operates continuously rather than in discrete events. Carbon supplied by plants fuels fungal metabolism. Sugars produced in leaves move downward through plant vascular systems and are released to fungal partners. In return, fungi supply phosphorus, nitrogen, and trace elements absorbed from soil volumes far beyond the reach of roots alone. The effective absorptive surface of a plant increases many times through this association. This partnership alters energy flow across ecosystems. A significant fraction of photosynthetically fixed carbon enters fungal networks rather than remaining within plant tissue. Estimates suggest that in some forests, up to 20% of plant-produced carbon is transferred below ground through mycorrhizal pathways. This carbon supports fungal growth and, indirectly, the wider soil community. Water transport follows similar routes. Hyphae conduct water along their length, moving it toward roots during dry periods. This transport relies on pressure gradients and cohesive forces within the cytoplasm. Although distances are small in absolute terms, the effect becomes significant when multiplied across extensive networks. During drought, plants connected to well-developed fungal systems often maintain higher water status than those without. This advantage arises not from water creation, but from redistribution. Fungal networks access moisture held in fine pores that roots cannot penetrate, extending the functional water pool available to plants. Material movement through these systems is directional but flexible. Resources flow toward areas of demand. A shaded plant receiving less light may receive more nutrients through shared fungal connections, while a dominant plant may export excess carbon outward. These transfers adjust dynamically as conditions change. Such redistribution influences competition and cooperation among plants. Seedlings connected to existing networks often establish more successfully than isolated individuals. Older plants may support younger ones indirectly through shared fungal pathways. The result is not uniform sharing, but conditional exchange shaped by local conditions. Beyond roots, fungal networks influence water retention at larger scales. By binding soil particles into aggregates, fungi increase pore stability. Aggregated soils resist compaction and maintain channels that facilitate infiltration. Rain enters more slowly and drains less abruptly, reducing surface runoff and erosion. These physical effects accumulate over time. Landscapes with extensive fungal activity often develop deeper, more structured soils. Water moves through them predictably rather than chaotically. Plant communities respond by establishing deeper roots and more stable growth patterns. Material movement does not stop at nutrients and water. Signaling compounds also travel through fungal networks. Chemical cues produced by stressed plants can propagate through shared mycelium, altering fungal behavior and influencing neighboring plants indirectly. These signals do not convey intention, but they transmit information through chemical change. The speed of such signaling is slow compared to animal nervous systems, but fast relative to growth processes. Changes can propagate over hours to days, aligning responses across connected organisms. This coordination emerges from shared pathways rather than central control. As these exchanges continue, the system remains in flux. Water availability shifts, nutrient gradients change, and biological demand varies. Fungal networks respond continuously, adjusting growth, transport, and interaction without resetting or restarting. This ongoing adjustment integrates the movement of materials across scales. Microscopic exchanges influence plant physiology. Plant responses alter litter input and root growth. These changes feed back into fungal activity, reinforcing or modifying existing patterns. Over time, the system develops characteristic rhythms. Wet and dry periods leave signatures in soil structure and community composition. Fungal species adapted to different moisture regimes rise and fall in prominence. The overall network persists even as its components shift. The continuity of these processes depends on living connections rather than static structures. When connections are broken, whether by disturbance or death, flow patterns change. New roots form as growth resumes. The system adapts rather than collapses. Attention now follows these material pathways further outward, toward the organisms that rely on them, and the transformations that occur as organic matter enters and leaves living systems. What moves through soil does not remain unchanged. It is altered, stored, and released again through processes that extend beyond exchange alone. As organic material enters the system, its movement follows a different pathway. Leaves, needles, roots, and wood fall or die in place, becoming part of the surface layer before gradually moving downward. Physical fragmentation by weather and animals increases surface area, but chemical transformation depends largely on fungal activity. Fungi capable of decomposing complex plant tissues initiate this transformation by releasing enzymes that target specific compounds. Cellulose is broken into glucose units. Hemicellulose yields a mixture of sugars. Lignin, one of the most resistant components of wood, is altered through oxidative reactions that few organisms can perform. These reactions require time and energy, proceeding slowly under most conditions. As decomposition progresses, material changes state. Solid structures lose rigidity. Carbon compounds are redistributed into fungal biomass or released through respiration. Nitrogen and phosphorus shift from organic to inorganic forms, becoming accessible to other organisms. Each step alters the chemical environment, influencing which species can operate next. Different fungi specialize in different stages of this process. Early colonizers exploit easily accessible compounds. Later species target remaining material that resists initial breakdown. This succession creates a temporal sequence within a single piece of organic matter, extending over months or years. Water availability shapes this sequence. In dry conditions, enzyme activity slows and decomposition stalls. In moist conditions, reactions proceed more efficiently. Oxygen availability also matters. In waterlogged environments where oxygen diffusion is limited, decomposition shifts towards slower pathways, altering carbon release patterns. As fungi digest material externally, byproducts accumulate in the surrounding soil. Some of these compounds bind to mineral particles, becoming part of stable soil organic matter. Others remain soluble and move with water, entering broader nutrient cycles. The balance between these fates determines how long carbon remains stored in soil. The presence of fungal hyphae influences this balance. Their physical structure protects some organic compounds from rapid breakdown by embedding them within aggregates. This physical protection extends residence time, allowing carbon to persist for decades rather than returning quickly to the atmosphere. At the same time, fungal respiration contributes carbon dioxide to soil air spaces. This gas diffuses upward and outward, linking underground processes to atmospheric composition. Although individual fluxes are small, their cumulative effect across ecosystems is significant. These opposing processes operate simultaneously. Storage and release occur together, regulated by environmental conditions and biological activity. No single outcome dominates permanently. Instead, soils oscillate between phases of accumulation and loss, shaped by climate, vegetation, and disturbance. As organic matter is transformed, it becomes part of a broader exchange system. Nutrients released by decomposition enter fungal networks and are transported toward roots or other microorganisms. In turn, plant-derived carbon continues to flow downward, sustaining further activity. This coupling maintains continuity between life above and below ground. Interactions among fungi influence these pathways. When multiple species colonize the same material, outcomes depend on competitive ability and enzymatic capacity. Some species inhibit others chemically. Some tolerate coexistence. The resulting community structure determines the pace and completeness of decomposition. Disturbance alters these dynamics. Fire, for example, removes surface organic layers rapidly, but leaves deeper material intact. Heat may kill some fungi, while others survive in protected zones. After disturbance, decomposition resumes under altered conditions, often favoring different species. Flooding introduces a different shift. Saturated soils reduce oxygen availability, suppressing aerobic fungi and favoring organisms adapted to low oxygen conditions. Decomposition slows, and organic material accumulates. When water recedes, activity increases again, releasing stored nutrients in pulses. Human activity influences these patterns through land use change. Tillage disrupts fungal networks physically, breaking hyphae and reducing connectivity. Fertilizer input alters nutrient availability, reducing plant dependence on fungal partners. These changes shift energy flow and material movement, often favoring faster cycling but less stable systems. Despite these pressures, fungal processes persist. Networks reform, species adjust, and functions resume under new constraints. The system adapts rather than stopping, although outcomes differ from those in less disturbed environments. As material continues to move and transform, the scale of influence expands. Processes that begin at the level of individual hyphae accumulate into soil profiles, ecosystem nutrient budgets, and regional carbon balances. Small reactions repeat countless times, producing large effects without central coordination. The movement of matter through these systems is continuous but uneven. Periods of rapid change follow periods of relative stability. Pulses of input and release alternate with slow accumulation. Fungal networks adjust to each phase, maintaining function across variability. Attention now follows these transformations further, toward how organisms of different sizes interact with fungal systems and how time cycles shape the pattern of activity across seasons and years. As decomposition proceeds, timing becomes a controlling factor rather than chemistry alone. The appearance of surface mushrooms reflects internal conditions that have developed gradually. Resource accumulation within the mycelium must reach a threshold. Water availability must be sufficient to support rapid tissue expansion. Temperature must fall within a narrow functional range. Only when these factors align does surface growth occur. The speed of emergence is a consequence of prior investment. Most of the cellular material required to build a mushroom already exists within the network. Expansion relies largely on water uptake rather than new cell production. Cells inflate, tissue unfolds, and structure rises quickly once internal pressure increases. This creates the impression of sudden appearance without sudden creation. The timing of these events varies by species. Some fungi produce fruiting bodies repeatedly throughout a growing season. Others appear only once every few years. These patterns reflect evolutionary strategies shaped by dispersal efficiency, competition, and environmental predictability. Rain often acts as the immediate trigger. Increased soil moisture restores internal pressure within hyphae and enables rapid transport of resources toward the surface. In dry soils, this movement is restricted. After rainfall, channels reopen and flow resumes. The response may take hours or days, depending on temperature and depth. Temperature modulates this response. Many temperate fungi fruit most readily between 10 and 20 degrees Celsius. Above this range, metabolic stress increases. Below it, enzymatic activity slows. Seasonal patterns emerge as a result, aligning reproductive events with predictable windows. Light plays a minor but measurable role. While fungi do not photosynthesize, light cues can influence the orientation and development of fruiting bodies in some species. These cues help position structures for effective spore release rather than energy acquisition. Once a mushroom forms, its lifespan is short relative to the organism that produced it. Days or weeks later, tissue degrades, collapses, or is consumed. The underlying network remains. Resources are reabsorbed where possible. Remaining material enters decomposition pathways, continuing the cycle. Spore release follows physical principles rather than intention. In many species, spores are ejected through surface tension changes at microscopic scales. In others, they fall passively or are dispersed by air currents passing through gills or pores. Each method reflects adaptations to local conditions. The quantity of spores released compensates for low success rates. Billions may be produced by a single fruiting event. Most will fail to establish. A small fraction encountering favorable conditions is sufficient to maintain population continuity. The environment into which spores land determines outcome. Bare mineral soil, fresh organic matter, or decaying wood, each present different challenges. Competition with existing microorganisms begins immediately. Growth proceeds only if resources can be accessed faster than rivals. This competitive landscape influences reproductive timing. Species fruit when conditions favor dispersal into environments where competition is reduced, such as after disturbance or during seasonal transitions. Timing thus reflects ecological opportunity rather than abundance alone. As these cycles repeat, patterns emerge across years. Some locations produce mushrooms reliably, others do so rarely. These differences reflect accumulated conditions below ground, shaped by water movement, organic input, and network development over time. The surface remains an unreliable guide to underlying activity. Absence of mushrooms does not indicate absence of fungi. Presence indicates alignment rather than dominance. Understanding this distinction reframes observation from appearance to process. These processes continue as organisms of different sizes interact with fungal systems. Invertebrates consume hyphae and fruiting bodies. Small mammals spread spores through movement and digestion. Larger animals alter soil structure, influencing water flow and aeration. Each interaction feeds back into fungal activity. Time cycles integrate these interactions. Daily temperature shifts influence metabolic rates. Seasonal changes reshape water availability. Multi-year trends alter species composition. Fungal systems persist through these cycles by adjusting rather than resisting. Attention now widens further, following how these living networks respond to disturbance and change, and how human activity intersects with processes that evolved long before human presence. Disturbance enters these systems in many forms, but its effects are rarely uniform. Physical disruption alters structure first. Compaction reduces pore space. Cutting or removal of vegetation changes organic input. Chemical changes follow as nutrient balances shift. Fungal networks respond by reconfiguring rather than withdrawing. When soil is disturbed mechanically, hyphae are broken into fragments. These fragments are not inert. Each retains living cells capable of growth. As conditions stabilize, new hyphal tips extend from cut ends, re-establishing connections within the altered environment. Recovery depends on moisture and temperature rather than on the size of the original network. Fire represents a different type of disturbance. Surface organic layers may be removed rapidly, while deeper material remains largely unaffected. Heat penetrates soil unevenly. Fungi near the surface may be killed, while those at depth survive. After fire, nutrient availability often increases temporarily as ash contributes minerals. Fungal species adapted to these conditions increase in activity. Flooding alters oxygen availability more than structure. Saturated soils restrict gas exchange, limiting aerobic metabolism. Some fungi tolerate low oxygen conditions for short periods. Others enter dormant states. When water drains, oxygen returns and activity resumes, often rapidly, as accumulated organic material becomes accessible. These responses illustrate a common pattern. Fungal systems absorb disturbance into their existing dynamics. Rather than resetting, they adjust. Species composition may change. Growth rates may shift. Functions persist, even as participants differ. Human land use introduces repeated disturbance. Agriculture, urbanization, and resource extraction alter soil structure, chemistry, and biological connectivity. Frequent tillage reduces mycorrhizal networks. Synthetic fertilizers reduce reliance on fungal nutrient exchange. These practices change the balance of processes rather than eliminating them. In managed systems, fungal activity often becomes more localized and less connected. Nutrient cycling accelerates, but stability decreases. Carbon retention declines. Water movement becomes less buffered. These changes influence long-term soil productivity and resilience. Despite simplification, fungal processes remain embedded. Networks reform after disturbance pauses. Species adapted to altered conditions replace others. Function continues under constraint rather than ideal conditions. Across landscapes, these dynamics produce variability. Adjacent sites differ based on history, management, and microclimate. Fungal systems reflect this variability, carrying traces of past use and disturbance within their structure and composition. At broader scales, patterns repeat. Regions experience cycles of wet and dry years. Vegetation shifts with climate trends. Fungal communities adjust accordingly, redistributing effort toward functions that remain viable. Some species retreat, others expand. Continuity emerges through change rather than resistance to it. These adjustments influence what is observed at the surface. Years with favorable alignment produce abundant fruiting. Other years produce little. These fluctuations reflect system state rather than success or failure. By the time surface structures appear, much has already occurred. Resources have accumulated, conditions have aligned, networks have adjusted. What appears briefly marks a moment of expression within a longer sequence of activity. As this sequence continues, attention moves outward again, from water and nutrients to organisms of increasing size, to the passage of time across seasons and years. The system does not pause between these scales, it operates across them simultaneously. The processes traced here do not conclude, they transition. What has been described forms the basis for patterns that repeat with variation, responding to climate, disturbance, and biological interaction. These patterns extend forward, shaping present conditions and influencing what comes next. Time introduces regularity without repetition. Seasonal cycles alter temperature, moisture, and biological demand in predictable ways, yet outcomes differ each year. Fungal systems track these changes through shifts in activity rather than through fixed schedules. Growth accelerates when conditions align and slows when constraints increase. In spring, rising soil temperatures reactivate metabolic pathways suppressed during colder months. Enzyme production increases. Hyphal tips resume extension. Organic material accumulated over winter becomes accessible as moisture returns and oxygen diffusion improves. These changes occur gradually, without a single starting point. Summer introduces different pressures. Higher temperatures increase metabolic demand while reducing water availability in surface soils. Activity retreats downward. Networks remain intact, but operate at reduced rates near the surface. Deeper zones continue functioning, supported by more stable moisture conditions. Autumn often brings renewed alignment. Cooling temperatures reduce stress. Rainfall increases soil moisture. Organic input rises as plants shed leaves and roots. Fungal activity responds by expanding again toward the surface, processing new material and redistributing resources. Winter slows visible processes without halting them entirely. In unfrozen soils, metabolic activity continues at reduced rates. Where freezing occurs, water becomes unavailable and enzymatic reactions pause. Hyphae survive these conditions through structural stability and dormancy. When thaw returns, continuity is maintained. Across years, these cycles leave cumulative effects. Soil structure evolves, organic matter composition shifts, species adapted to prevailing conditions increase in representation. Others decline. The system changes without losing function. Animals of increasing size interact with these processes. Invertebrates consume fungal tissue and spores, influencing population dynamics and nutrient movement. Their movement fragments networks locally while spreading propagules more widely. These interactions add variability without disrupting continuity. Small mammals transport spores on fur and through digestion. Larger animals alter soil structure through trampling and burrowing, affecting aeration and water flow. Each interaction modifies local conditions, influencing fungal response. Plants respond over longer timescales. Changes in fungal communities alter nutrient availability and water retention, influencing plant growth patterns. Vegetation shifts feed back into fungal systems through altered litter input and root exudation. These reciprocal adjustments integrate biological layers across time. Human presence introduces additional influences. Land use changes modify disturbance frequency and intensity. Urban soils experience compaction, altered chemistry, and fragmented organic input. Agricultural soils experience repeated mechanical disruption and nutrient supplementation. Forest management alters organic accumulation and species composition. Fungal systems respond within these constraints. Networks reform where possible. Species adapted to disturbance increase in prevalence. Functions persist, though outcomes differ from less managed systems. Nutrient cycling may accelerate while stability decreases. Modern research increasingly reveals these dynamics through molecular tools and long-term observation. Genetic sequencing identifies species previously indistinguishable by appearance. Isotopic tracing reveals nutrient pathways invisible to direct observation. These methods confirm processes long inferred from indirect evidence. Despite increasing understanding, much remains unresolved. The complexity of these systems resists simple prediction. Interactions vary by location, species composition, and history. What persists is the central role of fungi in linking material, energy, and time. Present conditions reflect accumulated processes rather than isolated events. The soil beneath current landscapes carries the imprint of past climates, vegetation, and disturbance. Fungal networks embody this history through structure and composition. As attention withdraws from individual mechanisms, the system appears less as a collection of parts and more as a continuous process. Movement occurs without announcement. Change accumulates without spectacle. Stability arises through adjustment rather than permanence. The surface remains intermittently quiet. Beneath it, activity continues, shaped by water, material flow, living connections, and time. What is visible remains brief. What persists operates beyond immediate notice. Human interaction with fungi began long before formal understanding. Early use was shaped by observation rather than explanation. Certain mushrooms were eaten without harm, others caused illness. Over time, patterns emerged, though causes remained unclear. This uneven familiarity contributed to caution rather than confidence. The unpredictability of surface forms reinforced this response. Mushrooms appeared and disappeared without obvious cause. Similar-looking species produced different effects. Some supported nourishment, others interfered with basic physiological functions. Without reliable markers, avoidance became a practical strategy. Despite this caution, fungi entered human systems in specific ways. Fermentation processes rely on fungal metabolism. Yeasts convert sugars into alcohol and carbon dioxide, enabling bread, beer, and wine production. These processes exploit controlled environments where outcomes remain consistent. Medicinal use followed a similar path. Observed effects preceded mechanistic understanding. The discovery of penicillin in the early 20th century revealed a fungal compound capable of inhibiting bacterial growth. This discovery did not introduce a new function. It revealed one that had operated within microbial competition for millions of years. Since then, additional compounds have been identified. Fungal metabolites contribute to immunosuppressants, cholesterol-lowering agents, and treatments for fungal and parasitic infections. Each compound reflects evolutionary pressures rather than human design. Chemical structures evolved to mediate interactions within complex environments. Research continues to expand this catalogue. Advances in genomics allow identification of biosynthetic pathways responsible for compound production. Cultivation techniques enable controlled expression of these pathways. What emerges is not a new system, but increased access to existing functions. Food production also reflects selective interaction. Certain fungi are cultivated for texture, flavor, and nutritional content. Others are excluded due to toxicity or unpredictability. These choices shape agricultural practices and dietary patterns without altering underlying biology. Industrial applications extend further. Fungi contribute to enzyme production for manufacturing processes. Their metabolic pathways support biofuel development, waste processing, and material synthesis. These uses leverage natural efficiencies rather than imposing external control. Throughout these interactions, constraints remain. Fungal growth depends on moisture, temperature, and substrate. Attempts to accelerate or suppress activity encounter limits imposed by cellular function. Systems respond to pressure through adaptation rather than compliance. Present conditions reflect accumulated choices. Urban soils differ from unmanaged ones. Agricultural systems prioritize yield over stability. Conservation efforts aim to restore connectivity disrupted by past use. Fungal responses vary accordingly, constrained by available pathways. Climate change introduces additional pressure. Altered temperature and precipitation patterns shift fungal activity and distribution. Some species expand into new regions, others retreat. Decomposition rates change, influencing carbon cycling. These adjustments occur incrementally rather than abruptly. Monitoring these changes requires long-term observation. Short-term studies capture variability, but not trend. Fungal systems integrate signals over years and decades. Responses reflect accumulated conditions rather than immediate events. As observation continues, the system appears less static than once assumed. Stability arises through continuous adjustment. Persistence depends on flexibility rather than resistance. Fungal networks exemplify this pattern through distributed function and local response. Attention now moves toward present conditions without resolution. The system continues operating. Water moves, materials transform, living connections persist. Human influence remains one factor among many, altering pathways without defining them entirely. The processes described do not conclude. They recede from immediate focus while continuing below. Surface signs may appear or remain absent. Either state reflects alignment rather than activity level. The ground remains active beneath present landscapes. What persists is not form, but process. Movement continues through time, shaped by conditions rather than intent. In present conditions, fungal activity continues under a wider range of influences than at any earlier point. Climate variability introduces shifts in temperature and precipitation that operate on shorter timescales, altering moisture availability and metabolic rates within soil. These changes do not replace existing processes, they modify their timing and intensity. In many regions, warmer winters reduce the duration of dormancy. Soil temperatures remain above freezing for longer periods, allowing enzymatic activity to continue through months that were previously inactive. This extended activity increases decomposition rates, altering the timing of nutrient release and carbon flux. Changes in rainfall patterns produce uneven effects. Extended dry periods suppress surface activity, while deeper networks persist. Intense rainfall events move water rapidly through soil profiles, transporting dissolved nutrients beyond shallow zones. Fungal interception moderates this movement, but capacity depends on network extent and continuity. Vegetation responds to these shifts, altering the quality and quantity of organic input. Changes in plant species composition influence litter chemistry and root exudates. Fungal communities adjust in response, favoring species capable of processing new substrates efficiently. In agricultural landscapes, these interactions are shaped by management decisions. Crop rotation alters organic input annually. Reduced tillage preserves fungal networks, while intensive tillage fragments them repeatedly. Fertilizer application changes nutrient gradients, reducing dependence on fungal transport while increasing leaching risk. Some systems retain partial connectivity. Fungal networks persist along field margins, beneath perennial crops, or within undisturbed soil layers. These remnants continue cycling nutrients and influencing water movement, even when surface activity appears simplified. Urban environments introduce different constraints. Soil compaction reduces pore space. Organic input is irregular. Chemical contamination alters microbial communities. Despite these limitations, fungi establish within parks, gardens, roadside verges, and beneath structures. Networks adapt to fragmented substrates, maintaining function at smaller scales. In forests, management practices influence accumulation and removal of organic material. Harvesting alters light penetration, soil moisture, and litter input. Fungal responses reflect these changes through shifts in species composition and growth patterns. Networks reorganize rather than withdraw. Across unmanaged landscapes, fungal systems respond primarily to climate and vegetation dynamics. Changes in species ranges alter network composition. New associations form, others weaken. These transitions occur gradually, layered over existing structure. Research increasingly captures these dynamics through long-term monitoring. Measurements of soil respiration reveal seasonal and interannual variability linked to fungal metabolism. Molecular analysis tracks changes in community composition without relying on visible structures. These methods confirm continuity beneath variable surface expression. Despite improved tools, prediction remains limited. Small changes in moisture or temperature produce disproportionate effects due to threshold responses. Interactions among species amplify variability. Outcomes depend on history as much as on current conditions. This uncertainty does not imply instability. It reflects sensitivity within adaptive systems. Fungal networks respond to local conditions, adjusting growth, transport, and interaction without requiring coordination across the entire system. Material continues to move. Carbon enters soil through plant input. Nitrogen and phosphorus circulate through decomposition and exchange. Water redistributes through pores shaped by biological activity. These processes remain active regardless of surface appearance. At any moment, portions of the network expand while others retract. Some hyphae grow, others age and degrade. Resources shift toward demand. This internal variability sustains function across changing conditions. Nothing here pauses. There is no baseline state. Activity reflects current alignment rather than equilibrium. What persists is process, rather than form. At the scale of individual organisms, these present conditions shape daily function rather than long-term direction. Hyphae respond to moisture gradients that shift within hours. A brief drying event alters internal pressure and slows extension. A return of humidity restores flow. These adjustments occur repeatedly without altering the identity of the network. Chemical conditions fluctuate in parallel. Nutrient concentrations rise and fall as water moves and as organisms absorb or release material. Fungal cells regulate uptake through membrane transporters, balancing internal demand against external availability. Excess is stored temporarily in vacuoles or converted into structural compounds. This regulation prevents abrupt failure during variability. Rather than maximizing growth continuously, fungi maintain capacity to resume activity when conditions improve. This restraint supports persistence across unpredictable environments. Interactions with other microorganisms continue to shape outcomes. Bacterial populations expand rapidly when simple compounds become available. Protozoa graze on both bacteria and fungi, releasing nutrients back into solution. These interactions modulate nutrient flow, influencing how much remains within fungal networks versus entering broader circulation. Competition remains local rather than absolute. Species overlap in function, but differ in tolerance. Some operate efficiently under low moisture. Others perform better under high nutrient availability. Shifts in conditions redistribute activity among these groups, rather than eliminating them entirely. At larger scales, these microscopic adjustments influence ecosystem-level patterns. Variability in fungal activity affects nutrient supply to plants, altering growth rates and competitive relationships. Vegetation responds through changes in root allocation and carbon export. These responses feed back into fungal systems through altered resource supply. In managed landscapes, these feedbacks are often constrained. Inputs and disturbances override some natural adjustments. Even so, fungal responses continue within available space. Networks adapt to impose schedules rather than disappearing. In unmanaged systems, variability accumulates more gradually. Extreme events introduce change, but recovery follows through reorganization rather than replacement. Networks retain continuity through fragments that survive disturbance and reconnect over time. Present conditions therefore represent a moving state rather than a fixed one. No single configuration defines normal function. Activity reflects current alignment among water, temperature, substrate, and biological demand. Observation at the surface captures only fragments of this activity. Fruiting events, when they occur, reflect local alignment rather than overall system state. Absence reflects misalignment rather than inactivity. Below ground, material continues to circulate. Carbon compounds move through decomposition and assimilation. Minerals shift between solid, dissolved, and biological forms. Water redistributes continuously through pores shaped by past biological activity. These movements do not converge toward a final arrangement. They remain open-ended, responsive, and contingent. The system persists by adjusting at multiple scales simultaneously. As attention remains with these processes, the sense of motion changes without stopping. Activity continues, but emphasis shifts away from individual interactions and toward their accumulation. What has been moving through soil, roots, and networks does not resolve into a final state. It spreads, redistributes, and persists. In places where conditions remain favorable, fungal networks thicken and stabilize. In others, they thin and fragment. Both outcomes represent continuation rather than decline. Material still moves, energy still flows, water still follows pathways shaped by biological structure laid down over long periods. The pace of change slows not because processes end, but because their effects become less distinct at short time scales. Day-to-day variation blends into longer trends. Individual growth events merge into background activity. The system continues without marking transitions. Surface signs become less frequent. Fruiting bodies may appear sporadically, or not at all. This absence does not alter the underlying state. Networks remain present, adjusting to conditions that do not favor expression above ground. Soil retains the imprint of past activity. Aggregates formed through fungal binding persist after individual hyphae degrade. Organic matter transformed earlier remains embedded within mineral structure. These remnants influence water movement and nutrient availability long after their origin. Roots continue to encounter these conditions as they grow. Their development follows pathways shaped by prior biological activity. Exchange continues where contact occurs. Where it does not, alternative roots form. The system does not depend on any single connection. Across larger areas, these patterns repeat with variation. Hill slopes, valleys, forests, fields, and urban margins each carry distinct configurations shaped by history and present conditions. Fungal systems operate within each, responding locally while remaining part of broader material cycles. Nothing signals completion. Processes do not conclude. They persist at rates that fluctuate with alignment rather than intent. What remains is continuity without emphasis. The ground continues to work quietly beneath present landscapes. Material enters, transforms, and moves on. Living connections form, adjust, and dissolve. Time passes without interruption. Under current conditions, fungal systems remain distributed across environments that differ widely in structure and constraint. In soils that experience minimal disturbance, networks expand in 3 dimensions, following moisture gradients downward and laterally. In compacted or fragmented soils, growth is redirected along cracks, root channels, and pockets of organic material. The form changes, but function persists. Water availability continues to regulate activity at fine scales. After rainfall, thin films of water coat mineral particles, creating temporary pathways for dissolved nutrients. Hyphae extend into these films, absorbing materials before they are displaced further downward. As soils dry, these pathways contract. Activity slows, but does not cease. The timing of these shifts matters. Short wet periods support surface level exchange. Longer periods allow deeper redistribution. Networks adjust accordingly, favoring zones that remain stable long enough to justify growth. This selective investment reduces energy loss and supports long-term persistence. Chemical gradients remain uneven. Nutrient-rich zones form around decomposing organic matter, root exudation sites, and areas of recent disturbance. Fungal growth concentrates in these patches, increasing enzymatic activity locally while reducing it elsewhere. This uneven distribution creates hotspots of transformation embedded within broader areas of lower activity. These hotspots influence surrounding organisms. Bacterial populations expand rapidly where simple compounds become available. Protozoa graze, releasing nutrients back into solution. Arthropods feed on fungal tissue, fragmenting networks while redistributing material. Each interaction modifies local conditions without disrupting continuity. Plant roots respond dynamically. Fine roots proliferate where fungal-mediated nutrient availability increases. Carbon export through roots adjusts to match return on investment. Where exchange declines, roots retract or shift growth elsewhere. This responsiveness integrates plant and fungal activity across changing conditions. In managed landscapes, imposed schedules intersect with these dynamics. Irrigation introduces water pulses that alter natural timing. Fertilizer application shifts nutrient gradients abruptly. Harvest removes organic input while disturbing soil structure. Fungal systems respond within the constraints of these interventions, maintaining partial connectivity where possible. Some functions become concentrated rather than widespread. Nutrient cycling occurs in remnant zones that escape disturbance. Carbon retention declines as networks fragment. Water buffering weakens where aggregation is reduced. These outcomes reflect altered structure rather than loss of biological capacity. Urban soils present additional constraints. Impermeable surfaces restrict water infiltration. Organic input is irregular and often imported. Chemical residues alter microbial communities. Despite this, fungi establish within green spaces, beneath pavement edges, and in unmanaged patches. Growth follows available pathways, adapting to discontinuous substrates. In forests, present conditions reflect layered histories. Past harvesting, fire, and climate variation shape current structure. Fungal communities reflect these histories through species composition and spatial arrangement. Networks persist through change by reallocating growth rather than maintaining fixed configurations. Climate trends influence these responses subtly and unevenly. Rising temperatures extend metabolic activity into periods previously inactive. Altered precipitation patterns change the frequency and intensity of wet and dry cycles. Fungal systems integrate these changes gradually, adjusting timing and distribution rather than shifting abruptly. Some species expand their range, others contract. Community composition shifts without eliminating function. Decomposition continues. Nutrient exchange persists. Water movement remains moderated by biological structure. Research increasingly reveals these dynamics through continuous measurement rather than isolated sampling. Soil respiration data shows variability tied to moisture and temperature rather than calendar time. Molecular analysis tracks community shifts without reliance on surface indicators. These observations confirm that visible absence does not equal inactivity. Despite improved resolution, prediction remains limited. Threshold responses amplify small changes. Interactions among species create non-linear outcomes. History shapes response as much as current conditions. Fungal systems operate through local adjustment rather than global optimization. At any moment, some parts of the network are expanding while others are contracting. Some hyphae transport resources, others degrade and are recycled. The organism persists through redistribution rather than stability of form. Material movement continues uninterrupted. Carbon enters through plant input. Nitrogen and phosphorus cycle through decomposition and exchange. Water redistributes through pores shaped by past biological activity. These processes remain active regardless of observation. There is no baseline state to return to. Activity reflects alignment rather than equilibrium. What persists is process rather than structure. Across different soil depths, activity varies in ways that are not immediately apparent from the surface. Near the top layer, organic input is frequent, but conditions fluctuate sharply. Temperature changes occur daily. Moisture rises and falls rapidly. Fungal growth here is episodic, responding quickly when conditions allow, and slowing when they do not. Below this layer, variability decreases. Moisture remains more stable. Temperature changes occur over longer periods. Hyphae operating at these depths grow more slowly, but persist for longer. Nutrient movement here influences plant roots that extend downward in search of stability rather than abundance. This vertical differentiation allows fungal systems to operate across multiple timeframes at once. Short-term responses occur near the surface. Long-term processes dominate deeper zones. Together, they integrate immediate conditions with accumulated history. Chemical transformation follows similar gradients. Fresh organic material is processed near the surface, producing compounds that move downward with water. Deeper zones receive these byproducts gradually, supporting organisms adapted to lower energy availability. Fungal networks bridge these zones, transporting resources across boundaries that would otherwise remain disconnected. In mineral-rich soils, fungi play a role in weathering processes. Organic acids released during metabolism interact with rock particles, slowly dissolving minerals and releasing elements such as calcium, magnesium, and potassium. These reactions occur over long periods, contributing to soil development rather than immediate nutrient supply. This contribution becomes more significant where physical weathering is limited. In stable landscapes, chemical alteration driven by biological activity dominates soil formation. Fungal involvement accelerates processes that would otherwise occur only through slow geochemical pathways. Interactions with plant roots influence this activity further. Root exudates alter local acidity and provide carbon sources that stimulate fungal metabolism. Increased activity enhances mineral dissolution locally, creating feedback loops between plant demand and fungal function. These feedbacks do not operate uniformly. Root distribution is uneven. Some areas receive concentrated input, others remain relatively inactive. Fungal growth mirrors this pattern, reinforcing spatial heterogeneity within the soil. As roots age and die, their channels remain. These spaces provide pathways for water movement and new growth. Fungal hyphae colonize these channels, extending influence into zones previously occupied by roots. This reuse of structure contributes to continuity across biological generations. The presence of these channels also influences gas exchange. Oxygen diffuses more readily along connected pores. This supports aerobic metabolism at depth, extending the zone where fungi can operate efficiently. Soil structure shaped by past biological activity thus influences present function. Over time, these processes shape the physical properties of soil profiles. Aggregation increases, porosity becomes more stable, resistance to erosion improves. These changes occur gradually and are rarely attributed to fungal activity directly, yet they depend on it. In environments where these processes are disrupted, soil properties shift. Compaction reduces pore connectivity. Water movement becomes less predictable. Nutrient loss increases. Fungal systems adjust by concentrating activity where conditions remain viable, but overall buffering capacity declines. Despite these constraints, continuity remains evident. Networks persist in fragments. Growth resumes when opportunities arise. The system adapts to imposed limits without abandoning its fundamental processes. Across landscapes, This adaptability produces diversity. No two sites operate identically. Each reflects a unique combination of substrate, climate, disturbance, and biological history. Fungal systems encode this information within their structure and activity patterns. At present, these systems operate under conditions shaped by rapid environmental change. Temperature trends, altered precipitation, and land use intensification introduce new combinations of stress and opportunity. Fungal responses integrate these factors without coordination, emerging from countless local adjustments. Some adjustments favor faster cycling, others favor retention. Outcomes vary regionally. What remains consistent is the role of fungi in mediating movement rather than producing endpoints. Material continues to enter soil through plant growth and decay. It continues to move through networks that adjust to current conditions. It continues to leave through respiration and transport. These flows define the system more than any visible structure. Observation captures moments within this movement, but not its continuity. Activity does not announce itself. It proceeds at rates determined by alignment rather than intention. At this point, activity reflects accumulated structure rather than immediate input. The arrangement of soil particles, pores, organic matter, and living tissue determines how new material moves once it enters. Fungal networks operate within this arrangement, responding to constraints created by earlier processes rather than starting from neutral conditions. When new organic material enters the soil, its fate depends on where it settles. Material resting on the surface experiences rapid temperature change and intermittent moisture. Material incorporated deeper encounters more stable conditions but slower access to oxygen. Fungal responses differ accordingly, with surface growth favoring speed and deeper growth favoring persistence. Enzymatic activity adjusts to these environments. Near the surface, enzymes operate in pulses, responding quickly to favorable conditions and slowing during stress. At depth, activity proceeds more steadily, limited by diffusion rather than exposure. Together, these modes allow transformation across uneven conditions. As decomposition progresses, intermediate compounds form. Some are rapidly consumed by other microorganisms. Others persist longer, binding to mineral surfaces or becoming incorporated into fungal biomass. These pathways influence how long carbon and nutrients remain within the soil system. Fungal tissue itself becomes part of this cycle. As hyphae age and degrade, their components enter the surrounding environment. Chitin fragments contribute to soil organic matter. Nitrogen bound within fungal proteins becomes available to other organisms. This turnover supports continuity without requiring permanent structures. The rate of this turnover varies. In favorable conditions, hyphal lifespan may be short, measured in days or weeks. In less favorable environments, structures persist longer, degrading slowly. These differences influence the balance between rapid cycling and long-term storage. Water movement continues to modulate these outcomes. Slow percolation allows interception and transformation. Rapid flow bypasses biological uptake, carrying dissolved material beyond active zones. Fungal networks reduce but do not eliminate this loss, acting as partial filters rather than complete barriers. At larger scales, repeated interception shapes nutrient distribution. Areas with extensive fungal activity retain more nutrients. Areas with limited connectivity lose more to deeper layers or runoff. These patterns influence plant growth and community composition over time. Roots respond to these gradients by allocating growth where returns are highest. Fine roots proliferate in zones where fungal-mediated nutrient availability is greater. This reinforces existing patterns, concentrating activity where networks are already established. These feedbacks produce stability without uniformity. Productive zones remain productive, less active zones remain less so. Disturbance can alter this distribution, but recovery often follows similar pathways unless structure is fundamentally changed. In systems where disturbance is infrequent, this leads to stratification. Layers develop with distinct biological roles. Surface layers process fresh input. Intermediate layers redistribute and store. Deeper layers retain legacy material. Fungal networks span these layers, linking them functionally. In systems with frequent disturbance, stratification weakens. Layers mix, connectivity declines. Processes compress into narrower zones. Fungal activity persists, but operates under tighter constraints, often favoring rapid turnover over retention. Despite these differences, the underlying mechanisms remain consistent. Enzymatic breakdown, absorption, transport, and redistribution continue. The form adapts to context, but the function persists. Across seasons and years, these processes integrate variability rather than resisting it. Dry periods reduce surface activity, but preserve deeper networks. Wet periods reactivate shallow zones. Extreme events introduce disruption, followed by reorganization. At no point does the system return to a previous state. Each phase builds on what remains. Past activity shapes present response. Present conditions influence future structure. Observation of mushrooms captures only brief alignment within this ongoing process. Their appearance signals sufficient internal resource concentration and favorable external conditions. Their absence reflects misalignment rather than inactivity. Below ground, activity remains continuous. Hyphae extend, retract, and reconfigure. Resources move toward demand. Material transforms through successive steps. These processes do not pause between observations. The system operates without central direction. Outcomes emerge from local interactions repeated across space and time. Stability arises through distributed response rather than fixed design. As conditions continue to shift, fungal systems remain governed by physical limits rather than preference. Diffusion constrains how quickly enzymes, nutrients, and gases move through soil. Water availability determines whether those movements can occur at all. Temperature sets the rate at which chemical reactions proceed. These constraints apply continuously, shaping activity without signaling change. Within these limits, fungi adjust allocation. Growth is directed toward zones where returns exceed maintenance costs. Areas that no longer provide sufficient resources receive less investment. Hyphae in those regions thin, fragment, or persist at low activity until conditions change. This selective maintenance reduces energy loss while preserving the option to resume growth. Such decisions emerge from local feedback rather than foresight. Concentration gradients guide transport. Mechanical resistance alters direction. Chemical signals from roots or decomposing material attract growth. No global assessment occurs. The system responds to immediate conditions, repeated across countless points. This repetition produces patterns that appear coordinated at larger scales. Nutrient-rich zones become more connected. Depleted zones remain sparsely occupied. These outcomes reflect aggregated local responses rather than overarching design. In soils with diverse substrates, Fungal communities partition resources. Species differ in enzymatic capability and tolerance. Some specialize in fresh plant material. Others target older, chemically complex compounds. Their coexistence extends the range of materials processed within the system. Temporal partitioning occurs alongside spatial partitioning. Some fungi operate primarily during cooler, wetter periods. Others remain active during warmer, drier conditions. These differences smooth overall system activity across seasonal variability. As a result, nutrient availability rarely drops to zero. Supply fluctuates but remains present. This continuity supports plant growth even under variable conditions, linking fungal persistence to broader ecosystem stability. At the interface between soil and atmosphere, exchanges remain moderated by biological activity. Respiration releases carbon dioxide upward. Moisture movement influences evaporation. Soil structure shaped by fungal aggregation affects gas diffusion. These processes connect subsurface activity to atmospheric composition without direct control. Over extended periods, these exchanges influence regional carbon balances. Areas with sustained fungal activity tend to store more carbon in soil organic matter. Areas with disrupted networks release carbon more rapidly. These differences emerge gradually, accumulating across years rather than appearing suddenly. Land management alters these trajectories. Practices that preserve soil structure support retention. Practices that fragment networks accelerate loss. Outcomes depend on frequency and intensity rather than single events. In landscapes undergoing change, fungal responses integrate new conditions incrementally. Shifts in vegetation alter input chemistry. Changes in hydrology modify water availability. Networks adjust distribution and activity accordingly. No clear boundary separates old conditions from new ones. This gradual adjustment complicates measurement. Short-term studies capture variability but miss trend. Long-term observation reveals persistence through change rather than replacement. Fungal systems embody this continuity through distributed structure. At present, these systems operate under increasing variability. Extremes become more frequent. Alignment windows shorten or lengthen unpredictably. Fungal responses remain adaptive, redistributing effort across depth, substrate, and association. Some networks contract, others expand. Some species decline, others fill emerging niches. Function persists even as composition shifts. Material continues to circulate through these pathways. Carbon enters, transforms, and exits. Nutrients move between organic and inorganic forms. Water redistributes through structures shaped by past biological activity. These flows define the system more than any momentary expression. Observation at the surface remains episodic. Fruiting bodies appear irregularly. Their absence conveys little about underlying state. Activity continues regardless of visibility. Below ground, growth and decay proceed together. Hyphae extend into new zones while older segments degrade. Resources are reallocated continuously. Persistence depends on redistribution rather than stability of form. These processes do not progress toward resolution. They remain open, contingent, and responsive. The system continues to operate within present conditions, shaped by physical limits and biological interaction rather than intent. At this stage, what governs activity most strongly is not novelty, but constraint. The system operates within boundaries established by physics, chemistry, and accumulated structure. Fungal networks do not expand indefinitely. They balance growth against maintenance, allocating resources where conditions allow continued return. This balance becomes clearer under prolonged stress. During extended dry periods, surface-level activity declines sharply. Hyphae near the surface lose water and reduce transport. Deeper networks remain hydrated longer and continue operating at reduced rates. When moisture returns, recovery occurs first, where structure remains intact. These responses depend on continuity rather than regeneration. Networks that remain connected resume function quickly. Fragmented systems require new growth before transport resumes. This difference influences how rapidly nutrients and water become available following stress. Chemical processes follow similar patterns. Enzyme production declines during unfavorable conditions, limiting decomposition. Intermediate compounds accumulate temporarily. When conditions improve, activity increases, and accumulated material is processed. This lag influences nutrient pulses rather than steady release. At broader scales, these lags shape ecosystem behavior. Plant growth responds to nutrient availability that reflects past rather than immediate conditions. This delay integrates short-term variability into longer-term trends. Fungal involvement in mineral interactions continues under these constraints. Organic acids released during metabolism interact with soil particles even at low activity levels. These reactions proceed slowly but persistently, altering mineral surfaces and releasing bound elements over time. Such slow processes become significant only when considered across years or decades. Individual reactions are small. Accumulated effects shape soil development and nutrient availability beyond immediate observation. Root systems respond to these gradual changes. Over successive growing seasons, roots explore zones where nutrient return remains reliable. Fungal networks reinforce these zones by maintaining transport capacity. Areas that fail to provide return receive less investment from both partners. This mutual adjustment contributes to spatial organization within soils. Productive zones persist. Less active zones remain marginal. Disturbance can disrupt this organization, but recovery often follows existing gradients unless structure is completely altered. At present, many systems operate under mixed influence. Legacy structure from earlier conditions interacts with current variability. Networks reflect both past alignment and present constraint. Activity represents compromise rather than optimization. Measurement of these processes increasingly relies on integration rather than snapshot. Flux measurements averaged over time reveal stability beneath variability. Molecular surveys reveal shifting composition without loss of function. These observations support the view of continuity rather than episodic activity. Despite increasing resolution, uncertainty remains. Local interactions produce outcomes that scale unpredictably. Minor differences in moisture, substrate, or species composition alter trajectories. This sensitivity does not indicate fragility. It reflects responsiveness. Fungal systems remain adaptable because they distribute function rather than concentrating it. No single point determines outcome. Failure in one area does not propagate globally. Activity reroutes through available pathways. Material movement continues under these conditions. Carbon compounds enter soil through plant input. Nitrogen and phosphorus circulate through decomposition and exchange. Water moves through pores shaped by biological activity. These flows persist regardless of surface expression. At no point does the system pause. Activity shifts location and intensity, but processes continue. Visibility remains unreliable as an indicator of function. The system remains fully engaged with present conditions, responding continuously to alignment among moisture, temperature, substrate, and biological demand. Within these remaining conditions, fungal systems continue to operate across uneven landscapes shaped by both gradual and abrupt change. Some soils experience repeated cycles of compaction and recovery. Others remain largely undisturbed. Fungal responses differ, but underlying processes remain consistent. Where compaction limits pore space, water movement becomes restricted. Oxygen diffusion slows. Fungal activity concentrates along cracks, root channels, and interfaces where gas exchange remains possible. Growth becomes linear rather than expansive, following available paths rather than occupying volume evenly. In contrast, loosely structured soils support more diffuse networks. Hyphae branch freely, forming dense meshes that intercept water and nutrients efficiently. These differences influence how quickly materials move and how evenly resources are distributed. Despite this variability, continuity remains evident. Networks adjust geometry rather than function. Transport persists even when routes change. Activity concentrates where conditions allow rather than attempting uniform coverage. Chemical gradients remain central to this adjustment. Areas enriched by root, exudates, or decomposing material attract growth. Depleted zones receive less investment. These gradients shift over time as inputs change and resources are consumed. The feedback between fungal growth and soil chemistry reinforces existing patterns. Active zones become more structured and retain resources more effectively. Less active zones remain less structured, allowing greater loss. These differences accumulate slowly, shaping long-term spatial organization. In systems where inputs are predictable, such as perennial vegetation, fungal networks stabilize around recurring patterns. Seasonal variability alters intensity, but not location of activity. In systems with irregular input, activity shifts more frequently, following transient opportunities. These dynamics influence how ecosystems respond to stress. Systems with stable fungal connectivity recover more quickly after disturbance. Systems with fragmented connectivity respond more slowly, requiring regrowth before function resumes. At present, many landscapes exhibit mixed characteristics. Some areas retain deep connectivity, others remain fragmented. Fungal systems operate across these gradients, maintaining function where possible and adapting where constrained. Biological diversity within fungal communities supports this adaptability. Functional overlap allows processes to continue even as species composition shifts. Loss of one species is often compensated by others capable of similar activity under different conditions. This redundancy contributes to resilience without uniformity. Outcomes vary locally, but core processes persist. Decomposition continues. Nutrient exchange occurs. Water movement remains moderated by biological structure. As these processes continue, they influence above-ground patterns subtly. Plant growth reflects nutrient availability shaped by fungal mediation. Species composition responds to these differences. Vegetation patterns change gradually rather than abruptly. These changes feed back into soil systems through altered litter input and root behavior. Fungal networks respond accordingly, reallocating growth and transport. The cycle continues without reaching a fixed endpoint. At present, observation captures only fragments of this cycle. Measurement techniques reveal trends, but not complete dynamics. Understanding remains partial, reflecting the distributed nature of the system itself. What remains consistent is the absence of finality. Processes do not resolve. They adjust continuously, integrating new conditions into existing structure. Persistence arises through ongoing response rather than stability of form. The system remains active beneath current landscapes. Material moves, energy flows, biological connections form, shift, and dissolve. These processes continue without emphasis or closure. Across the full range of present environments, fungal systems continue to operate without reference to boundaries humans tend to impose. Property lines, land use categories, and surface features do not align with the pathways that matter below ground. Networks follow moisture, substrate, and opportunity rather than designation. In transitional zones, such as forest edges, grasslands meeting cultivated fields, or urban margins, fungal activity often intensifies. Organic input varies. Root communities overlap. Disturbance is intermittent rather than constant. These conditions favor flexibility over specialization, and networks reflect this by maintaining diverse growth patterns. In such zones, resource movement becomes less predictable. Nutrient inputs arrive irregularly. Water availability fluctuates sharply. Fungal systems respond by spreading risk. Growth is distributed across multiple substrates rather than concentrated in one. Transport routes remain flexible, able to redirect as conditions change. This flexibility is supported by internal organization. Cytoplasmic flow within hyphae allows rapid redistribution of resources. Storage compounds accumulate when supply exceeds demand and are mobilized when conditions reverse. These internal buffers reduce dependence on continuous external input. Metabolic rates adjust accordingly. During favorable periods, respiration and enzyme production increase. During constraint, activity slows without ceasing entirely. This modulation allows persistence through variability rather than maximization during brief alignment. Interactions with plants continue to shape these responses. Root exudation changes with plant developmental stage, stress level, and environmental conditions. Fungal growth mirrors these changes, expanding toward active roots and retreating from senescing ones. This responsiveness integrates plant life cycles into soil processes. In perennial systems, this integration produces long-term stability. Roots and fungal networks co-develop over years, establishing persistent exchange pathways. In annual systems, integration is more transient, reforming each cycle. Both modes support function under different constraints. Animal interactions remain part of this system. Soil invertebrates alter structure through movement and feeding. Their activity fragments networks locally while redistributing organic material. These effects introduce heterogeneity without eliminating continuity. Larger organisms influence conditions indirectly. Grazing alters plant input. Trampling changes compaction. Burrowing modifies pore structure. Fungal systems respond to these changes by adjusting growth location and intensity rather than abandoning affected zones. At present, these interactions occur under increasing variability. Shifts in climate introduce new combinations of moisture and temperature. Extreme events occur more frequently. Alignment windows shorten or lengthen unpredictably. Fungal systems respond through redistribution rather than resistance. Some zones experience accelerated cycling as higher temperatures increase metabolic rates. Others experience suppression due to moisture limitation. These differences create spatial variability that influences broader ecosystem patterns. Carbon movement reflects these shifts. In some areas, increased decomposition releases carbon more rapidly. In others, constrained activity promotes retention. Outcomes depend on local structure rather than global trend. Nitrogen and phosphorus cycles adjust similarly. Availability fluctuates with microbial activity, water movement, and plant demand. Fungal mediation smooths but does not eliminate variability, maintaining continuity without uniformity. These processes operate simultaneously across scales. Microscopic reactions accumulate into soil profile changes. Local interactions influence landscape-level patterns. No single scale dominates. Function emerges from integration rather than hierarchy. At present, the system remains open. Inputs continue. Transformations occur. Outputs leave. No endpoint defines completion. Activity reflects alignment among conditions rather than progression toward a state. Observation remains partial. Surface indicators capture moments rather than processes. Measurement integrates signals over time without resolving all interactions. Understanding remains provisional, reflecting the system's distributed nature. What persists is movement. Material enters soil. It is transformed, redistributed, and released. Biological connections form, adjust, and dissolve. These processes continue beneath present landscapes without interruption. Across all of these present environments, what persists most clearly is the absence of a stable reference point. Conditions fluctuate, inputs vary, and interactions shift. Yet fungal systems continue operating through redistribution rather than correction. There is no attempt to restore a previous state. Activity aligns with what is available. This alignment depends on proximity. Hyphae respond to immediate surroundings rather than distant conditions. Moisture detected at one tip does not guarantee availability elsewhere. Transport within the network redistributes resources, but growth remains local. This balance between local sensing and system-wide redistribution defines how fungi persist across heterogeneous environments. The geometry of networks reflects this balance. Branching patterns change where resistance increases. Growth angles shift in response to gradients. Sections encountering persistent limitation reduce activity, while others expand. Over time, this produces irregular but functional structures adapted to local constraint. These structures influence how future material moves. Water follows established pores and channels. Nutrients encounter interception points shaped by earlier growth. Roots follow paths of lower resistance created by past biological activity. The system carries forward its own history. This history is not static. As conditions change, some pathways lose relevance, while others gain importance. Networks adjust by reinforcing useful routes and allowing others to degrade. This selective persistence supports continuity without rigidity. In soils with long, uninterrupted histories, these pathways become deeply embedded. Connectivity remains high. Resource movement is buffered against short-term variability. In soils repeatedly disturbed, pathways are shorter-lived. Networks rebuild frequently. Function persists, but stability declines. Present conditions often combine both patterns. Legacy structure interacts with recent disruption. Fungal systems operate within this mixture, maintaining function where continuity remains and adapting rapidly where it does not. The role of fungi in these systems is not to optimize outcomes, but to mediate movement. Carbon compounds are transformed and redistributed. Nutrients are intercepted and exchanged. Water is retained and channeled. These functions continue regardless of surface expression. As these processes continue, they influence what becomes visible above ground without controlling it directly. Plant growth reflects nutrient and water availability shaped by fungal mediation. Vegetation patterns shift gradually. Surface changes lag behind subsurface adjustment. At present, this lag becomes more pronounced under rapid environmental change. Surface indicators respond slowly to subsurface restructuring. Fungal systems integrate new conditions before they are reflected above ground. This integration does not resolve uncertainty, it accommodates it. Variability becomes part of the operating environment rather than a disruption. Fungal networks persist by remaining responsive rather than resistant. Material continues to circulate through these pathways. Organic input enters soil. It is processed, redistributed, and either retained or released. Each cycle modifies conditions slightly, contributing to longer-term change without discrete transitions. The system remains active at all times, even when surface indicators are absent. Activity shifts location and intensity, but processes do not stop. Growth, degradation, transport, and exchange continue within constraints set by present conditions. There is no point at which these processes complete. They do not converge toward balance or closure. They continue as long as material, moisture, and energy remain available in some form. As attention lingers on these processes, the importance of mushrooms becomes apparent without needing emphasis. Their role is not defined by visibility or scale, but by continuity. What they do is not occasional. It is constant, distributed, and foundational. Without fungal activity, material would accumulate rather than move. Dead plant matter would remain largely intact. Nutrients bound within complex structures would stay unavailable. The circulation that supports life on land would slow to a point where many systems could no longer function as they do now. The soil itself would change. Aggregates formed through fungal binding would weaken. Water would move more abruptly, draining quickly or pooling without moderation. Roots would encounter more resistance and fewer pathways. Growth would become more dependent on immediate input rather than stored capacity. Plant communities would reflect these limits. Access to phosphorus and nitrogen would decline in many environments. Cooperation between roots and fungi would be absent, reducing effective reach into soil. Competition would intensify where resources remained accessible, altering vegetation patterns over time. Carbon movement would also shift. Decomposition would slow unevenly. Some material would persist longer, while other compounds would be lost rapidly through less efficient pathways. The balance between storage and release would change, affecting atmospheric exchange over long periods. The absence of fungal mediation would not result in sudden collapse. It would produce gradual constraint. Systems would simplify. Diversity would narrow. Stability would decline as fewer pathways remained available for adjustment. In human systems, the effects would be equally distributed. Medicines derived from fungal compounds would not exist. Fermentation processes that rely on controlled fungal metabolism would be absent. Many industrial enzymes would be unavailable. These outcomes reflect lost access to functions that evolved long before human use. What makes fungi essential is not any single contribution, but the way they connect processes that would otherwise remain separate. They link dead material to living systems. They connect water movement to nutrient availability. They integrate plant activity with soil structure. They operate across time scales that exceed individual lifespans. Their persistence does not depend on stability of form. Networks fragment and reconnect. Species shift. Structures appear briefly and disappear. Function continues through redistribution rather than permanence. This way of operating shapes the environments in which other organisms exist. It creates conditions that allow complexity to persist without requiring coordination. It supports variability without collapse. It enables change without erasure. Mushrooms, as surface expressions, reflect moments when internal conditions align. They are not the work itself. They indicate that growth, exchange, and accumulation have reached a point where reproduction becomes possible. Their appearance marks continuity rather than origin. When they are absent, the work continues without display. Networks remain active. Material moves, energy flows. The system does not wait for recognition. The world shaped by these processes does not announce their presence. It carries their influence in soil texture, plant distribution, water behavior, and nutrient movement. These effects accumulate quietly, becoming part of the background against which other life operates. As focus gradually loosens, what remains is not an object or organism, but a set of ongoing processes embedded in the ground. They continue beneath forests, fields, cities, and unmanaged land alike. They persist through variability, disturbance, and time. Nothing concludes here. Activity does not resolve. It continues, shaped by conditions rather than intent, operating without pause beneath the surface. What becomes clear as this view widens is that fungal systems do not support life by directing it. They support it by making movement possible. They reduce friction within the living world. They allow matter to change form without leaving the system entirely. In soils where fungi remain active, nothing stays isolated for long. Carbon does not remain fixed in dead tissue. Minerals do not stay locked inside rock or wood. Water does not move without resistance or guidance. Each element encounters pathways shaped by prior biological work. These pathways persist beyond the organisms that form them. When hyphae degrade, the structure they influenced remains. Pores stay open, aggregates hold, channels continue to guide roots and water. Present conditions are shaped by activity that occurred long before. This persistence explains why fungal influence is often underestimated. Their work is not visible in motion. It is visible in what no longer happens. Nutrients do not disappear as quickly. Water does not rush away as abruptly. Systems do not fail under moderate stress as easily. Where fungal activity is reduced, these absences become noticeable. Soils lose structure. Nutrient loss accelerates. Plant growth becomes more dependent on constant input. Variability produces sharper consequences. The system narrows in how it can respond. The importance of fungi is therefore not dramatic. It is cumulative. It emerges from repeated small actions that alter the conditions under which other organisms operate. Over time, these alterations define what is possible. In natural systems, this allows diversity to persist. Different species occupy overlapping roles without direct competition, eliminating one or the other. Resources circulate through multiple routes. Failure in one pathway does not halt the system. In human systems, this same capacity underlies reliability. Food production, medicine, and material processing depend on fungal functions that evolved without reference to their later use. These functions are accessed rather than created. What links all of these outcomes is scale. Fungal systems operate at scales too small to see directly and too large to comprehend as individuals. Their influence emerges only when viewed across space and time together. Mushrooms briefly make this influence visible. They rise, release spores, and decay. Their presence draws attention, but their absence does not indicate inactivity. The processes that produce them continue before and after without interruption. As the focus softens further, the system resolves not into an image, but into continuity. Beneath varied landscapes, similar processes unfold. Material moves, networks adjust, conditions are integrated rather than resisted. There is no final state toward which this movement tends. There is only persistence through change. As long as organic material enters the ground, as long as water moves through soil, as long as living systems exchange matter, fungal activity remains embedded within those flows. The ground does not rest. It works quietly, shaped by networks that form, fragment, and reform. Their influence remains even when they are no longer present in the same configuration. What remains at the end of this movement is not a conclusion, but an ongoing condition. The world above continues to depend on processes below that operate without recognition, without pause, and without resolution. As this movement continues, what remains most consistent is not any particular structure, but the conditions that allow structure to form at all. Fungal systems persist because they remain responsive to what passes through them. They do not store meaning, they store possibility. Across soils shaped by different histories, the same processes repeat with variation. Organic material enters. It is broken down, redistributed, and incorporated. Water moves along paths shaped by earlier growth. Nutrients circulate through living connections rather than remaining isolated. These actions do not depend on awareness, they depend on contact. The importance of mushrooms and of the organisms that produce them lies in this quiet mediation. They allow the ground to remain workable rather than inert. They keep resources moving rather than locked away. They support systems that must adjust continuously rather than remain fixed. Nothing here requires visibility. Fruiting bodies appear when conditions align and disappear when they do not. The work continues regardless. Networks expand and retract. Material is transformed and passed on. Time moves through these systems without interruption. What exists above ground rests on this activity without reference to it. Forests, grasslands, fields, and cities all depend on processes occurring beneath them that operate without pause. These processes do not announce themselves. They persist through repetition. As focus settles, the system does not close. It remains active, shaped by current conditions and by the accumulation of past activity. There is no final configuration. There is only continued movement through soil, through networks, through time. Thank you for listening to the Sleepy Mind channel. I hope you have enjoyed spending time with the mushrooms.